Welcome to this episode of Lectures with Liddington. Last episode, we talked about mitosis, which is a type of cell division that we use when we need to make new cells. So for growth or repair. In this episode, we're going to talk about meiosis, which is a different kind of cell division, which not all cells are going to go through. It's a very specific process. So why? Why would a cell undergo meiosis instead of mitosis? And the answer is really, well, what are we making that cell for? In mitosis, we're looking at growth and repair, replacing old cells that are damaged or just making some new ones if you're physically growing. In meiosis, we're not trying to replace cells. We're trying to make entirely new organisms. Meiosis is a process that we use when we are trying to make sex cells. And this is specifically sperm or eggs. So here is mitosis, and then over here is meiosis. This is the process. At the end here, each one of these cells represents either an egg or a sperm, depending on whether we're talking about in a male or a female. And these are what we call sex cells. Sex cells, or sperm and eggs, are what we call haploid, which means they have half of the genetic information. So they're going to have half the number of chromosomes as the parent cell. So down here, see 1, 2, up here, 1, 2, 3, 4. So that's haploid because it has half of that genetic information. Ultimately, what we want to do is to take a sperm and an egg and unite them together so that we can make a new organism, whether it's a new human, a new turtle, a new frog, whatever it is. So a zygote is that single cell that results when a sperm and an egg unite. That zygote is going to be what we call diploid. Or double. So it has the full set of genetic information. So a little practice here. This is a diploid cell because it has a full set of genetic information. This end product of mitosis is also diploid. It has 1, 2, 3, 4. 1, 2, 3, 4. Same number of chromosomes. Whereas in meiosis, my parent cell has 4. And those daughter cells have 2. So they are haploid. They have half the genetic information. What this does is it creates genetic diversity. Down here, I have cells that are genetically different than what I started with before. I'm going to end up with half of the information from mom and half the information from dad coming together in that zygote. And as we continue to talk through this process, we'll see a couple of other ways where we'll get some variation in genetics. But our end game, the why behind meiosis, is creating new organisms using these haploid sex cells that have genetic diversity. So what does this look like? Well, ultimately here, this is my starting point. Here is my cell in interphase. It's just doing its thing. It has the genetic information. And then it undergoes DNA replication during which phase of the cell cycle? Aha, yeah, right, S phase. That's when we synthesize a new copy of DNA. So we replicate that DNA in order to prepare for this cell division. Well, instead of it going through mitosis, it's going to undergo meiosis. So what happens is meiosis actually happens in 2 phases, 2 divisions. So what I do is I start with meiosis 1. These two chromosomes line up, and when they line up, what's going to happen is a phenomenon called crossing over, where we'll have the two pairs line up at the middle, and then pieces of those two different sister chromosomes will exchange genetic information. So notice here we've got a little piece of that one chromosome that has swapped places with the other. This is called crossing over. And what this does is it mixes up what possible genes can be inherited together. So for example, if we had hair color and eye color on this chromosome, we might have blue and blue over here, or blue and brown. You wouldn't have blue hair. Blue eyes and blonde hair might be over here, and then brown and brown on this side. All right, well, now we've swapped it. So we now have the possibility of inheriting brown hair and blue eyes together, or brown eyes and blonde hair. So we have this mixing. So this is another area where we can increase our genetic variation. So this is still making a diploid cell, because ultimately this will divide and I'll have, two. Because this is my chromatid. My chromatid has the genetic information. Right, so 1, 2, 1, 2. I need to drop down again. So what I'm going to do is undergo a second round of division called meiosis 2. At this point, what I'm going to be separating aren't those, we call them homologous. So these are homologous chromosomes. So I'll separate homologous chromosomes. Whereas in meiosis 2, I'm going to separate those chromatids. So I'm going to now separate those two different sides. I've lined my chromosome up in the middle, and I'm going to separate, I'm going to pull away the two different sides. So this will have my one chromosome, and this will have the other. Notice, I started out with 2 chromosomes, and now each of my daughter cells only has 1. Haploid. I have half the amount of genetic information. So this is what happens during meiosis. Sometimes things can go wrong. We call these chromosome mutations. Inversion and translocation. So these are 2 different types of chromosome mutations, and these can happen in either mitosis or meiosis. So either cell division, these can happen in. Non-disjunction is a separate, and we'll talk about that in a second. So in inversion, what happens is I take a piece of a chromosome and basically it flip-flops itself in a different direction. So you'll end up with sort of a mixture of information, which can have a big impact or not so much, sort of depends on what's happening. So notice here the wideband has moved up. So now the wideband is here. They're just in a different order. This could make a small difference. It could make a huge difference. It depends on what information is present in that section. Translocation, location, right? I'm changing the location. So this piece of chromosome, boop, is now over here. I'm swapping on different chromosomes. Inversion is within the same chromosome. Translocation is between different chromosomes. Now, non-disjunction, this is a situation that only occurs during meiosis because of the way those separations occur. What happens in non-disjunction, so if we break the word down, junction is a connection, right? So disjunction would be breaking a connection. Non-disjunction does not break a connection. So that might be breaking it down a little bit, and it might be more confusing. But basically what happens is, at one of the divisions, things don't divide properly. So here, this is meiosis 1. What'll happen is these homologous chromosomes don't separate properly. And then they both stay in the one daughter cell, and the other one gets nothing. So what's going to result is two empty daughter cells, two daughter cells that are missing that genetic information completely. And then I'll end up with two that have extra copies. These cells will not survive. They do not have enough genetic information to survive. So whether it's a sperm or an egg, they will not survive with that much missing genetic information. Over here, what I end up with is an issue in meiosis 2, where I won't have a disconnect. Over here, I end up with the right amount. I have haploid because this one separated properly. Only the one separates improperly, so I don't separate the sides. So I end up with two copies. What happens now is this sex cell, when it unites with another, it will create what we call a trisomy, which means it will get 3 copies of that chromosome, because the other sex cell will come in with just 1 copy like it's supposed to, and it will have 3 copies. Here, this would be what's called a monosomy. And like I mentioned before, generally this isn't going to survive. A zygote here will not survive because it doesn't have enough genetic information. Sometimes a trisomy can survive. And one of those examples is called trisomy 21. And that's what we call Down syndrome. So that is one example of a situation where a non-disjunction will result in a successful zygote, but you can actually see the impacts of having that extra genetic information. All right, so let's pause here and do a little practice.